My name is Kaitlyn Miranda. I am a PhD student in the Elsa Flores Laboratory, and I'm here at the Cancer Engineering Summit. I am presenting my poster on investigating modulation of ΔNp63 and MDM2 to therapeutically target p53 in cutaneous squamous cell carcinoma. So previously in our lab, it was shown that in thymomas, in p53 null thymomas, if you knock down ΔNp63, which is an oncogenic isoform of the protein p53, that you see an upregulation in amylin. And this upregulation in amylin causes a decrease in glycolysis and an increase in apoptosis. So we wanted to recapitulate this effect in vivo, where we are repurposing a diabetes drug called Pramlintide that is a synthetic analog of amylin to lower ΔNp63 levels, decrease glycolysis and increase apoptosis. So we wanted to move this to squamous cell carcinomas because they are characterized by having high levels of ΔNp63. So we have done experiments in our cell lines, and we have seen an interesting phenotype where some cells are sensitive to Pramlintide, meaning that their glycolysis is lowered, and some cells are resistant. And we were also able to see a similar effect in vivo, that there was a response to Pramlintide in cutaneous squamous cell carcinoma, but there was a lot of variability. So we did a genome-wide CRISPR screen to identify sensitivities of and identify mechanisms to further sensitize these cells to Pramlintide. And because our lab is interested in the p53 family, we wanted to look at MDM2, which was a hit on the pathway. Now, MDM2 is an inhibitor of p53, so we want to essentially inhibit MDM2 to give p53 its tumor suppressive qualities back. So we do this through a variety of mechanisms by siRNA knockdown of MDM2 in combination with Pramlintide, as well as looking at a clinically available small molecule drug that inhibits MDM2 called Nutlin. And we were able to see this synergistic effect where Pramlintide and Nutlin cause cell cycle arrest and decrease proliferation in our cells. We were now able to move this to an ex vivo model where we quantified proliferation via EDU, and you can see the green is EDU positive cells, and that with Pramlintide and Nutlin treatment, there is a significant decrease of of cell proliferation, both shown through imaging in our microtumor models and through flow cytometry. So our next steps is to move this in vivo, and our goal is to locally deliver both Pramlintide and Nutlin to mouse models that have cutaneous squamous cell carcinoma via microneedle patches.